Hey, Hayden. So James didn't show in the Zoom meeting to set it up. Is it possible to? Um,
Um, it's recording to his cloud automatically. Um,
okay.
but I think that he's the only one that can start the live stream, so it may just not be live streamed.
Okay.
Well, let me see.
Just to be sure, the last class was recorded or not?
Yeah, I believe it was.
Okay.
Let me check. I have the option to live stream to YouTube, but I'm pretty sure that that will just take me to a page where like I have to sign in. But
All
I'll
right.
try. I'll try in a second. Um, yeah, last week's episode. Last week's class was live streamed. So let me try here. Yeah, it takes me to a sign in page. So I guess this week's, unless James logs in to set it up, we won't be live streaming this week.
Is there a way to remove transitions from all slides?
Transitions or animations?
Transitions.
I'm not sure.
Because it's pissing me off this slow fading out of the previous slide and fading in of the new slide. It takes like 2 seconds to change a slide. Um, None. Let me see now. Okay, I think I did it. Okay, that's much better. All right, I'll set it up here. It's 4:10. We should be starting already, but I will probably wait 5 more minutes because there is another guy, Shane. Um, he came the first two classes, so he is about 50% of the class. So I think I'll wait 5 more minutes to just in case he's coming. Um,
Oh, I'm sorry. Are you talking about me?
oh, you are here. All right.
Yeah, yeah,
So
yeah.
he is in the Zoom meeting.
Yes, yes.
All right. Okay. So I'll screen share. Let's make this work. All right. And all right. So I have a new student. I don't know you. So what is your name and what is your background? All right. I'm a physicist too. I'm James' student, and James cannot teach the class today. So we are still deciding how intense the lecture will be. But the first few lectures, we will cover the basics about chemical reactions and using tellurium. You guys are not restricted to doing that. You can, yeah, you came for the first class, right? And you didn't come last week. All right. So last week we covered central dogma of cell biology. Are you familiar with this? All right. Metabolic networks, protein signaling networks. Let me get rid of this. And can I make it? Who cares? Signaling protein, protein signaling networks, basic mechanisms behind them, gene regulatory networks, and how to. So we learned notation, ODE representation, and we implemented them in Tellurium to see their behavior. So the time series of the species, basic motifs, and I explained intuitively what is the law of mass action last class. Are you familiar with that? The law of mass action. So I will quickly show to Nick in the whiteboard what is the law of mass action. And the guys in Zoom, both of them, sorry, saw this. So let me make sure. Yeah, so here we have Shin and Niraj, they saw that. So I'll just quickly explain to Nick in the whiteboard. Okay, maybe we don't have a. All right, let me use my infamous paint whiteboard. So we have, let's say, a reaction A plus 2B goes to C. Okay, so I have a molecule A combines the two molecules of B, producing molecule C. So when we ask ourselves what is the rate at which this reaction happens, you're losing A, so you have a negative rate. You are losing B, you have a negative rate for B, but you have twice the rate of B because you need two molecules of B per molecule of A. But here we are assuming that they are reacting. One molecule of A is reacting with two molecules of B. So for that to happen, you need to have the molecule A and two molecules of B at the same position in the same time, the collision. So the probability of a collision to happen is proportional to the concentration of the molecules. And so the law of mass action is basically saying that the rate of this reaction V, let's say, is a rate constant K, and it's proportional to the concentration of A, and it's proportional to the concentration of B squared because you need two molecules. So probability is independent. So the probability of have two molecules of B is the probability of have one times the probability of have another one. So you're multiplying all probabilities. So this is your rate constant. This is not true for all cases. This is only true if you are assuming that your solution is an ideal gas. If it's not an ideal gas, let's say you have for example, large molecules, and they occupy a lot of space, maybe the law of mass action will not apply. But this is true for these most of these biochemical reactions like metabolism, sugar, blah blah blah. So it's the assumption that the rate is proportional to the concentration of the species. And there is a derivation for that using the grand canonical framework, starting with the Gibbs energy. And I will not go through this, but I will just show you more or less how it looks like. And you guys will tell me if you want me to go through this in detail in another opportunity, next class, for example. So basically here we assume equilibrium. So delta G is 0. Then we have the chemical potential for a solution, and then we combine those, and then the algebra goes, and eventually you arrive that the equilibrium constant of that reaction is the product of all concentrations, and they are exponentiated by their stoichiometric coefficient. So you can derive that mathematically. So you guys tell me if you want me to go through this derivation in the next class. But today we will learn about another type of network. Just get to know it, which is the physiologically based pharmaco-kinetic network model. So we learned the gene regulatory, we learned the protein signaling, and the metabolic networks. So this is a network that is more like for the entire body. So you transform the entire body into a network. And so you have the lung, the brain, the blood vessels, the kidneys. You are taking in a drug, you are eliminating a drug, and this drug spreads throughout the entire body, and different drugs will have different penetrations in different tissues. So you want to ask, for example, what is the concentration of the drug in the brain if you want the drug to go to the brain, or if you don't want that drug to go to the brain. You need to consider that this drug is traveling throughout the entire body with different transportation rates, for example. And we use the same ODE systems to model the transfer of concentration between different compartments. So you have like drug, and let's say you have drug in the lung, you have a variable DL, then you have the concentration of drug in brain, so you have another variable DB, and so on and so forth for all the compartments. So if you are developing, let's say you are using protein signaling network to study the effect of a drug on cell cycle, let's say. But you also have to consider that the concentration of the drug inside the cell is an outcome of all of this. So if you want an appropriate model for the individual, you might have to consider a PBPK network. So in these types of networks, the nodes represent the organs, and they have different volumes. So by having different volumes, you can hold different amounts of material. So let's say you have your blood. It's a large volume, and then you have, for example, you want to target a tumor, which is a very, very little volume. So the loss rate of this drug from the tumor to blood will be large, because the blood is such a big compartment. So you have to consider the volume of the nodes if they act as sources or sinks. So for example, your gut can be acting as a source if you are eating something. So let's say you're modeling a metabolic network and you are eating sugar, your gut works as a source, your kidneys could work as a sink, etc. So besides that, so that was a very quick introduction about PBPK. Let's go back to what we were doing last time, which was working with Tellurium. And this is a very straightforward framework to getting the ODEs for a reaction. So if you have those stoichiometric coefficients, the rate of change of a species will be basically the difference of the stoichiometric coefficients before and after. So you have, for example, for A, you have the stoichiometric coefficient of the product, AP, minus AS, and the same for the other species. These multiplied by the rate V, but the rate V, if we assume law of mass action, it becomes, yeah, this is an example. If we assume law of mass action, we have the stoichiometric coefficient exponentiating the concentration of the species. And for reversible kinetics, you have the same thing, but you have two reactions, a forward reaction and a reverse reaction, back and forward. And in this case, you just subtract both. So you have, in one reaction, we have the forward direction. So we have each stoichiometric coefficient exponentiated by the, each concentration exponentiated by the stoichiometric coefficient. Then you subtract the other reaction the other way around. So you have the stoichiometric coefficients of the products exponentiating the other term. So this is the final framework. For species A, this is the difference of both stoichiometric coefficients, and then the forward reaction is this one. So Kf is one of your rate constants. Then A to the stoichiometric coefficient times B to the stoichiometric coefficient. These from the left side of the reaction and from the right side of the reaction, the same thing, but with the stoichiometric coefficients of the products. Make sense? So this first parenthesis stands for the it's the ratio of mass from sources to products. And the second parenthesis stands for the law of mass action assumption. So let's try to do this one. So we have this reaction. It's a reversible reaction and build the ODs. That's our exercise to make sure we understood what's going on. So I'll give you guys 5 minutes to do this one. So for the derivative of A, what did you get, Niraj and Shin? Let's see. So you're doing 3 minus 7 times. So you need different VA. So what is V times? Yeah, so you need different rate constants for the forward and the backwards reaction. Make sense? So it should be V1 here, let's say. V1, and then you have A. So you rotate times. Do you mean power like this? So are you saying okay? Yeah, so okay, makes sense. So you have A to the 3, then B to the 4, and then C to the 5. Oh, okay. I forgot this. C to the 5. Correct. Then minus V2. And then it's A to the 7 times B to the 2 times C. A to the 7 times B to the 2 times C. Perfect. That's it. And then analogous for B. That's correct. Make sense? Oh, forward and back. Okay. Yes. Yeah. So the idea is there are two reactions happening. So you divide them. Let me take a look. Sorry. Yeah. So this is the wrong arrow. It should be just let me see. Oh no, this is a second one. Ignore that. So we're just doing the first one. Yeah. So let me cover this. So you have two reactions. So you would be writing two ODEs for A, let's say, but then you subtract them. All right. Shin. Shin, did you follow?
Yeah. I got the same equation as Niraj.
Okay, sure. Let's not do the second one because I think if we got the first one, the second one is I mean, do you guys want to do the second one? Do you know how to do the second one?
Like just adding all the equations together.
Yeah, you have to be careful that you have B in two different equations. So how do you handle that? You cannot have two ODEs for B, right? Yeah, you just add all of them together. Yeah. What is that clear for you guys?
Yes.
Yeah, so you just add all of them together. So I think we're good with this one, all right? So if this problem comes up to you again later and you have trouble, we can come back to it. All right. So whenever you see a reaction like this, and there is this K above the arrow, this K generally in textbooks refers to the constant that is multiplying our law of mass action. So it's not the rate, it's the constant of the rate. The rate depends on both the constant and the species. So you have to be careful with this. Because sometimes we write a V above it, and then we say V equals K times A to the 2 times B. But when you see a reaction like this and it says K, generally it stands for the constant that multiplies the rate constant, not the rate itself. We saw a little bit about steady state. I hope you all are familiar with it. The steady state is when all your derivatives is zero, which means nothing is happening anymore. If all your derivatives are zero, your system stalled. And that's different from equilibrium. Chemical equilibrium is when you don't have transfer of matter anymore. So it's different from the steady state. You can have steady state without chemical equilibrium. For example, in a back and forth reaction like this, you still have a reaction going on, so you are not reaching equilibrium. But you can reach the steady state nonetheless. Just to be clear about that. And the chemical equilibrium for very large reactions, well, you just use the law of mass action for everything, and you can reach an equilibrium constant. We did that last class, which is basically the ratio between the forward and backwards reaction. And this equilibrium constant tells you how much of the products you will have compared to the sources. And the meaning of this is how stable are your reactants or your products. So if your equilibrium constant is a large number, it means you have a stable product. And if your equilibrium constant is very low, you have a more stable reactant. So basically the difference of stability between your reactants and your products tells you how much of each you have. When you reach the steady state, so you set all your derivatives to zero, that's how you find your fixed points. So later we'll do like stability analysis of these networks. So we have to find the fixed points. That's the first step. You set all your derivatives to zero and find what are the concentrations of each species in the long term. So let's do this for this one, this simple reaction. It's different from the other reactions we did last class, because we have a source for A. So we start with A at zero, and we have a source for A. What is the ODE corresponding to this reaction? And well, just answer the questions. But the point we want to reach is, what is the steady state concentration of A? Did it? Right, that's it. Let's see our other guys. K1 over K2. That's right. Shen, how about you, my friend?
Yeah, it's a K1 over K2.
What is the shape of A?
The shape of A, like as the time changes. I mean,
Yeah,
that depends.
how does it depend with time? What is the shape of the curve?
That depends on the initial amount of A.
Yes, it depends on the initial amount of A. Let's say A is zero. A starts at zero.
then it's increasing and it converges to that K1 over K2.
Perfect. Shen, agree?
Yep.
Sorry, this was Shen. Niraj, Niraj, do you agree? I'm confused. Yes, okay. Let's go. So you can solve the equation analytically. It's, as you said, it's an exponential. It's like the capacitor charging. Um, um, yeah, so starting at a certain value, as you change one of the parameters and you'll have different steady state solutions, and they all follow this exponential, and it tends to K1 over K2. Yeah, I guess that's very simple to us. Scanning the initial value doesn't change the steady state. We saw in the last class, we saw a case where setting different values, the initial values, you have different steady states. In the case of A goes to B, B goes back to A. If you start with different concentrations of A and B, you arrive at different steady state solutions. But here you don't. It doesn't depend on the initial state. So when you care, for example, about homeostasis, do you all know what homeostasis is? Homeostasis, Chen?
I'm sorry, what?
What
I
is
didn't catch.
homeostasis?
You
Uh,
try
Huh?
so you try to reach the equilibrium state, sort of, or not the equilibrium state, but you have a predefined state. You try to go to that state is called homeostasis.
Good. Yes. So for living beings, exactly as Niraj said, you have a state you want to be, a healthy state. And as you perturb the state, you want to go back to your default state. And this is one of the systems that have these characteristics. So if you make a perturbation, it goes back to the same value as before. So this is a characteristics you look for when you are studying, for example, when you need homeostasis for your system, when you need stability. We'll do this parameter scan in Tellurium because we need parameter scans to do our projects later. And so implement this ODE in Tellurium and set these parameters. This is our exercise. Do you have access to a computer or something, Nick? I don't know if this one will work, but I hope it will. I will help you. So I will go to my collab thing and do that with you guys. So have you implemented anything in Tellurium so far? Yeah, okay, good. Well, I have to install stuff first. Right. So you know how to load it and build, okay. So this is what you want to implement in your little Tellurium code. It takes so long to install it. Okay. All right. So let me do this guy here. It's so tell me when you get something like what I have here. You can also copy what I did if you don't want to stress too much about it. Give me a thumbs up when we're good to go. Niraj, do you want to see the analytical derivation of the law of mass action X class?
Yes, if other people are also interested, I would say.
Okay. Important. Oh, you don't have it installed? Did you install it? I do have it installed. Important. Then it goes to for A. Like it. Oh, so this is I see, yeah. I see. No. It's you're using the call instead of I don't know the name of this shit. Is this one.
Colon, I see.
Column, yes. Oh.
And what is the one? Yeah.
Yeah, I don't know if the comma works.
Yeah, evidently it doesn't. I was just,
You have
I was checking I see the colon because otherwise I don't know. So
Okay.
you have to,
And then you can do a plot array.
there we go.
All right. Shen,
Yes.
thumbs up.
Yeah. Thumbs up.
Do you want to see the full derivation of law of mass action next class?
Yeah, I'm interested. Yeah.
What about you, Nick?
Sure, if you want to, I. Did it before.
Okay. You've seen it? Oh, okay. So maybe you can do it. No, I'm joking. I mean, if you wanna do it, you can do it.
Sure.
Okay.
Do it.
If you're up to the challenge, we can organize it. All right. Just if you're interested. If you're not, I mean, I will do it. All right. Next step. Next step is doing the parameter scan. All right. So first, when you plot it, and here in the slide, we were supposed to use the plot thing, but from the previous example from last class, we were already using the plot array. The plot array thing. So if you do, just let me comment this out and do, and I think I have to do this. And then R2.plot. We get the same thing, but to manipulate many runs of our simulation, we'll use the plot array function. For now, because I want to follow the slides thoroughly, I'll be using the plot for now. All right. So one thing we want to do is learn how to change the values of a species in our Python code. So we can do, and also so you have your model object. In my case, it's R2, but it could be anything else for you. If you do .a and you say .a is 10, now your initial value of A is 10. So if you run this again, your A starts at 10 and goes to 1. So do that by changing your variable value in the Python part. Give me a thumbs up if you got it.
So does that mean that you have a value initial set to 10, right?
Yes.
So, but we can directly do that in the, t.load a.
Yeah, but when you want to run many simulations, you want to update your variable like from the outside. Otherwise, you have to manipulate the variable inside the string. We don't want to do that.
Okay, right, makes sense.
So you want your model as a black box, and then operate your model from the outside.
Right, right.
All right. Next, we'll try this thing here. So you simulate, and here he's doing 100. You simulate, you plot, and then you simulate, and you will plot again and see what happens. And someone please explain to me what is going on. Why when I run it the second time, it gives me a different result?
Basically because the value where the first plot takes the value of A, the next plot takes it from there on.
Yeah, that's it. So it starts from the last value of A. So it simulates your simulation sequentially, not from the beginning. If you want to simulate it from the beginning, we need to use a command. And this command is, so this is explaining what is going on. So first you load a model and then you simulate. Your model object gets to a certain state, and then if you keep simulating it, it starts from where it was left. So if you want to simulate again from the beginning, which is what we want to do for a parameter scan, then you need to reset your parameters. So here we have 3 ways of resetting. We'll cover the first 2. So r.reset resets the parameters. Sorry, resets the variables. So it sets A to A0, And reset all resets both variables and parameters. So if you run the simulation, your A is different. And let's say you change the K of your rate. So you set K1 to 2, and then K1 to 4, and then K1 to 5. And then you want to reset everything, including the parameters, to the default parameters and default initial conditions. Then you use the reset all. So let's try resetting the simulation using your model object R, or whatever you named it, dot reset, before running it a second time. So I have R2 plot, and then I will do R2 dot reset, and then simulate again and plot again what happens. I'm still getting two different results. Is that wrong? What is going on here? Yeah, I'm resetting the value to back to 0, because that's what I specified in the model string. Yeah, that's it. Clear? Thumbs up. Shen?
Yeah, yeah.
All right.
Yeah,
Let's
I just don't know where the thumbs up button is.
Okay. So now let's change a parameter. So let's instead of doing a equals 10, let's do k1 equals 10. And then reset. If I do it, nothing happens, right? Because I'm not resetting the parameter. The function reset just resets the species. If I want to reset the parameter 2, then I use the reset all. Like this. Then when I do this, I get a different result. See, this is 8, this is 0.8. Okay. All good? So try that, get the same result as me, and we move on. And now we'll start doing our parameter scan. So a parameter scan, you are basically looping over a certain parameter and then running your simulation from the beginning again and do many different values of that parameter and seeing what's going on. So we build a loop over our parameter here. Let's say here we are looping over our initial concentration, our A0 parameter. So we can do in, and then we use this NumPy function arrange from 0 to, it has to be a float, so it has to be 0.0 to 4.0, and then 0.5 by 0.5, that's our interval. And then we do what? First, reset. Otherwise, you will run the simulation from where the previous simulation stopped. So we'll do our r dot reset. Then we simulate it, and then we plot. And what happens when I do this is I get many plots for my different parameters. So that's not the appropriate way of doing it. The right way is building doing a plot array, but then we don't show the plot array. We use plot array with a modifier show equals false, and we just show it after the entire loop. So then it will be te.plot array. And then it will show not quite. So here what I have to do is, let me see. My result is my simulation. Oh, I'm doing, this is why. I have to get rid of this. Okay, I'm getting an error. Plot, okay, so I have to say plot result. Oh, let me see what's going on here. Something is not right. Oh yeah, I'm not updating the variable. After I reset it, I have to update the initial value of A. So A is A0, and now it's correct. All right. So please get this result, or more or less this result. And of course, we can use more clever labels. For example, this can be doing this. Yeah. Yeah, it doesn't. How do I set color? Color. Oh. All right, I'll figure it out later how to set a different color. Did you all get something like this? Thumbs up? Miraj? All right, good.
Yep.
How did you set the colors?
So I need to define the color first. So, okay, I'll just copy paste the code maybe.
Huh?
I had defined the value of color, then I've
Okay.
give the color in that variable.
Can you paste that in the chat?
Yeah, sure.
Oh, was the next slide. Reset. There it is. And what you did was, okay, so you do okay. A 0 times 2. All right. Also, it is color, the attribute. All right. Yeah, I never seen this. So you're doing color is, and then a string C. What is C doing? Oh, that's nice. So I have to import matplotlib. Oh yeah.
Uh, no, you don't need to import matplotlib.
Okay. All right, and then I do color. What's color? Hopefully this is right. Very nice. All right, good. Next. So this is just showing how to do the labels. I like to do the labels like this using a format string. But you can also add strings together, but I don't like using this str command. But that's non-important. Cycling through colors, you can also use this command, reset color cycle false. But I think it's more beautiful doing the way that Niraj did. What if I don't multiply by 2? It's a different palette. Okay. Yeah. So I think it's better to do it this way. And even maybe if I divide by something, can I divide by 2? To get a... It's not a... Okay, it's not a... It converts... Oh, it's converting to an integer. Does it need to be an integer? Yeah.
Mm,
Yeah. Because sometimes when we want a very graded parameter scan, and then we want, for example, something from red to blue, all hues, continuously. But there are other ways of doing it. All right. Now let's scan our parameter K1 between 0 and 2. And then K2 between 0 and 2. Mm-hmm. How are you all doing? Okay. All good? Oh, so you mean this? Or without this? Is this what you mean?
Yeah.
But is it? Oh, down here. All right. Okay. I feel like an old man learning Python. Okay. You all got this? Shen?
Yep.
All right. So now we'll get to the hill functions, and we will elaborate on why the hill functions are the way they are and why we use them like that. And we'll start by studying these enzymatic reactions. So an enzyme, as for example, a phosphorylation or dephosphorylation or ubiquitination enzymes, those molecules, they are catalysts. They grab a certain molecule and they make a change at that molecule. So you don't waste your enzyme, but you are converting a source to a product. And there is an intermediary stage where your enzyme is binded, I don't know, to your source protein. So we are explicitly modeling this intermediary stage. Why? Well, because if this binding site is unstable, you have a lower reaction rate, so you generate less product. And if you have a, for example, very fast enzymatic reaction, then you don't need a very strong bond between the enzyme and the source protein. So that's why we're modeling explicitly. And this system, in the end, will give us the hill function we use. So I will draw this schematically.
Just one doubt in the previous plots.
Mhm.
So if you go to those plot, yeah. So the first plot, the x-axis is going from 0 to 5 only, but we are simulating for 100, right?
Yeah, but yeah, so the first one I'm simulating to 5 and the other one I'm simulating to 100. Yes.
Okay, you're only simulating. Okay, cool. Okay, my
Yeah, I do it here. Dot simulate 0 to 100.
right. Okay, cool. Yeah, makes sense.
All good?
Yep.
Or let's do this. So someone draw me a diagram of this reaction taking place. And by the way, where is the
Come up with a molecule?
yeah, the molecule. Yes. A representation of the molecule. Like those diagrams that you see in Wikipedia, you know. Yeah, someone stole the pencil. And it wasn't me, although I am a Latino. I have to specify that it was a joke, otherwise James will get mad at me later. Quick diagram, how is it? Let's see.
Quick diagram.
That's nice. Yeah.
There we go.
Yeah, we don't have this step though in the reaction. Right.
I mean, you effectively have some kind of alteration process which is internal to the
Yes,
ES.
you have that, but that diagram would correspond to let's say, so you have this, let's it's reversible. And then you have this, let's say. What you draw would be this. So the reaction would be we would have a extra step like this, right? Yeah. Makes sense? All right. Yeah. So this diagram would correspond to this reaction right here. So the diagram that represents the reaction we have is this one. Let's say here we have another one, another thing. But that works, no problem with that. Did you say that it's reversible? All right. Okay, that's it. So what is this? This could be a phosphorylation, like you're adding a phosphate group to a protein. This could be a dephosphorylation. It could be a GTP GDP conversion or GDP GTP exchange or ubiquitination.
Couldn't be phosphate. Because you'd have to have a phosphate.
If you're ignoring the existence of the phosphate, it's this. So it's a minimalistic model that represents a very large set of reactions that happen inside the cell, let's say. Sure, yeah. So if you had a phosphate, you would have something like I don't agree. I think this is the correct representation. What would be how would you modify the ODE to?
Have it's in a kinase.
Yeah.
Phosphate. And usually what those do is that they either they have the phosphate group on them or they're able to connect to another protein or amino acid that's been phosphorylated and pulls it over and connects on and then pulls it off and then decomposes. I mean, technically though, you either have an alteration of like an enzyme can't do that because an enzyme has to be immuted, like it's not changed, immutable in the process, right?
Yeah. But so you're saying that the kinase is not being recycled in the reaction?
No, the kinase can be recycled. The difference is that the kinase, it can or can't, right? It sometimes it's not. Just depends on whether or not it's phosphorylated.
So what he's saying is, if our enzyme is a kinase, it's losing the phosphate group, so we will also have a change in the enzyme itself. So it can be recycled or not. In this reaction, we are assuming it's being recycled. So let's say this protein, this kinase, is abundant and always being recovered. So you don't care about the loss of concentration, the concentration fluctuations of your kinase, for example. Then this would apply. But yeah, I get what you're saying. Yeah, so this is considering the following assumptions. First, your enzyme is not suffering from the reaction. A kinase would. A GTPase would too. But for example, a ubiquitin ligase could be this one, could be this reaction, but again, it needs another participant, the ubiquitin, to bind to the protein. So if you're using this to model ubiquitination, you would include the ubiquitins. But again, if you don't care about the ubiquitins because they're so abundant, then I think this applies. So it all depends on whether the assumptions hold for the system, right? All right. And what are we supposed to do here? So first, the total amount of enzyme is the same in these model. So if you do ES plus E, it's conserved. And yeah. Next. These are the ODEs that represent the system. So you have a forward and backwards reaction. That's why you have minus KF and then plus KR, because you're gaining matter from the ES side. Same for S. So E and S have these back and forth. And then P, you can only produce P out of this. So we have to be careful because we might generate infinite amounts of P if we have regeneration of S in the system. If you just start with a constant amount of S, you can only have a constant amount of P. Right? Make sense? All right, so let's implement this in Tellurium. And this is the first cool system, cool reaction where we will find some interesting behaviors. So I'm looking forward to this one. And in these examples, it's so clear how Tellurium is appropriate to reactions, because writing the reactions is so much easier than writing the ODEs. So Tellurium, what it's doing is basically converting the language of reactions to the language of ODEs. So you just need to be careful about setting the correct rates, reaction rates. Mm. So give me a thumbs up when you get this result. In the meanwhile, I'll go get some water for me. All good? All done? All right. Nick is done. Niraj, Chen.
Yeah, cool.
Chen? All right. Okay, all done. Good. So what is going on here? I'm losing all my S. S is being converted all to P. And then I have a like an interval of time where some of my E is converted to ES in the transition between S to P. But in the long term, I'll recover all my E and lose all my ES because, well, S goes to P. So this is a weird way of saying S goes to P, right? It's a more complex way of saying S is going to P, right? I just saw S in the bathroom, by the way. Yes. It's a weird, this model right there, it's a weird way of saying that S goes to P. Like this. So right? Did you see it? So S goes to P is the simplest model you have of this, and it will have more or less the same behavior, like losing S and gaining P. But doing it like this will get us other behaviors other than these simple exponentials, right? So to see these other behaviors, so this is the result, by the way. You can also plot E total. If you say here, E total is, then you have to do column equals E plus ES. Let's see if it plots. No. Maybe I have to specify. No, I'm not gonna do this. It's too dirty. So if we change, for example, the initial amount of S, a very, very high S, very high amount, initial amount of S. What is the behavior you expect? Do you expect these shapes to change? And then run and verify. What do you expect? You already run, by the way, right? Does it make sense? What is the difference?
Well, really, I mean, I was playing around to try and see how big of a problem I could get. for
Okay.
each.
Mhm.
But in doing that, I changed the yeah, it looks like it kind of gets rate limited by the amount of heat
Mhm.
overall. But say something more like that.
Mhm. So E becomes the limiting factor. And in this case, you get a constant generation of P and a constant loss of S. Make sense? If you want a big bump, I guess you just do this. So you need a strong forward reaction, a weaker backwards, and weaker generation of P. Yeah. Make sense? Yeah. for a big bump. So as I increase KF, I get a bigger and bigger bump.
Yeah. Initial derivative. Initial slope.
Yes, exactly. Yeah, I like to think of like matter, transfer of matter. So I am transferring a lot of matter to ES and not so much out of ES. So I get a big reservoir of matter in around ES. That's how I think of it. This. Yeah. Because you could have a strong derivative in the beginning, but also a strong derivative to the generation of P. So if I like here, I have 100 for KF, but if I have also 100 for R, I will not get a strong bump. So I need to retain that matter in yes. All good? So okay, let's get back to the exercise, but that's an interesting idea. So starting with s equals 10, as Nick said, we'll have a limited reaction. So we have too much s for too little e. So in the end, what happens is I have this constant loss of s and constant generation of p for like after 1 hour, let's say, up to 10 hours, and then it goes back to exponential behavior. Why? Well, because eventually I have similar amounts of S and E, then it goes back to the previous behavior. Make sense? How can I analytically demonstrate that I will have a linear regime using the ODEs when I start with high S, high amounts of S? I'm going to gym to do that, by the way. So we have a linear regime here and here, right?
Because ES itself is relatively small in that region.
No, but you cannot look at the graph. You're saying I have high amounts of S. Okay. How do you demonstrate that you have this linear regime?
So we can see in the OD itself that ds by dt, the ES value, we can ignore the complex ES form as compared to the minus KF dot E dot S. So that can show us that it would constantly decrease.
Okay, so you're basically taking this reaction and ignoring this term and this term.
Yep.
For just the first one, what about the
No,
second?
just for ds by dt, where S is constantly decreasing, linearly sort of.
So, but when you ignore this value and consider just this value,
Right.
you still have an exponential decrease, right?
All of the E is going to be transferred into ES effectively. So for S itself, that term is negligible.
So you have a low amount of E, so this cancels this first term. Hmm. Mm. And then only the second term is being considered here.
Of course,
And then okay, so you're assuming that this is 0, this is 0. So you have these two and this.
doesn't work though, because that's the E.
And we can also approximate ES to be just E, since all the enzyme would be basically converted to ES.
Yes,
Then,
it's E.
yeah, we can substitute ES to be just E. Then we can have a linear
You mean
decay.
E0?
Sorry?
You mean E0 or E?
No, the complex ES, we can substitute complex ES by just E because we have excess of substrate. So the limiting step would be just the enzyme. So ES formed would be equal to E itself. So ds by dt would be equal to, but yeah, that would increase. No.
I don't think that would work because E plus ES, sorry, because E and ES are fundamentally different. They are anti-correlated.
Hmm.
Really what you want to do is that you want to first show that change in E is about 0, right? Change in E is 0, then you can take that first term, the derivative of E, that has to be this KR times ES plus RES. ES itself is also like the inverse of E in some sense, 1 minus E, right? So if E is constant, then that 1 minus E is constant, and therefore
Yes.
this is
Yes, that's it. That's it. Yeah, that's it. So what Nick arrived at the solution. So basically what is going on here is that S is very high. I think you missed one argument. So I will try to rebuild it and then to explain to them, and then we see if everything is all right. So S is super high. This means that the first reaction is super fast. So we're basically, by saying S is very high, we are saying the first reaction is super quick. So it reaches the steady state much sooner than the others. With this argument, we can say the derivative is 0. Go ahead.
it doesn't reach steady state.
But so what the argument is, the first reaction is much faster than the others.
Oh, you mean in the sequence? Okay, yeah, in the sequence, yes. Okay, sorry.
Yes, I mean,
Sorry.
I might not be explaining it right, like effectively. Okay, no problem. So the first reaction is super fast. If the first reaction is super fast, it reaches a quasi-steady state. It's not a steady state. It's a quasi-steady state where this derivative is negligible compared to the others. So by doing this, we can say dE by dt is 0, which means E is constant. If E is constant, ES is constant because E plus ES is constant. So if E is constant and ES is constant, we plug those into the S reaction, and there we have that ds by dt is a constant rate. And then we'll have the same for dp by dt. We'll have that derivative is also a constant rate. And then we can demonstrate that at a certain point we have a linearity. Is it clear? Niraj and Shen?
Sort of clear.
Okay. So many people do this. Look at this. So whenever a reaction is very fast, so you have dx by dt equals a bunch of stuff. Whenever this is very, very fast in your system, you can do an approximation, which is saying that this derivative is zero because you are in a quasi-static regime. Because this reaction is very fast, you just wait a little bit. This system, the concentration of this X molecule becomes constant. So, and whenever the system changes a little bit, X immediately catches up. And then by doing this, then X is constant. In our case, we have ES plus E is a constant because we are conserving the total amount. So if one is constant, the other is constant. And then from this argument, you will derive that S decays at a constant rate and P grows at a constant rate.
Yeah,
Is that
makes
okay?
sense. Yeah.
Right. You might be thinking, well, but S is varying, right? Is varying. S is varying. So this is not exactly constant. Yeah, it's not exactly constant, but when you assume this, you plug this into the first equation, and then you have E times S equals a constant. So that first term must be a constant. Obviously, this is not true for the entire system. I mean, you will find those arguments like, but we're losing S and we're gaining P. So it doesn't hold for the entire system, but that's not the point. The point is that this is a behavior that works when S is very high. As you lose S, you lose your argument, and then the system will not behave that way anymore. Good? People in the Zoom, Shen, is it okay?
Yeah, I guess.
Yeah, all right. We'll see. By the way, they do this a lot in papers to reduce the number of reactions. So we will go back to this eventually. All right. So this is the behavior you should expect, and that's what I have here. Here he's going to 5. So if I want more or less the same behavior, I go to 5 like this. Yeah, that's the same graph. Okay, good. Quasi-steady state reactions. That's the name of the slide. All right. Now let's see what happens when I have a very low production of P. Well, unfortunately, the slide already shows the solution, but so ideally we want give me a gas, Chen. So by looking at this reaction, I have this reaction here, and I have a very slow generation of P. So basically this derivative here
First graph. First time initially, that's relatively fast.
yeah.
Second grade is gonna just have to sort of sync for ES over time.
Yes. So if you think quasi statically, basically you have a steady state that updates as you exactly. That's it. That's a good interpretation. So let's do that in the code. Yeah, sometimes I have what?
Quasi-steady.
Yeah, quasi steady state. Yes. Yeah, that's what I'm getting. Like implementing Tellurium seems dumb when you understand the equations, but Yeah, but sometimes why is this different than oh, it's 0, that's 0. Okay. All right. So after you understand, it seems dumb to so then plug the number and see exactly what you thought, right? But arriving at these conclusions take some energy, and then after you understand, you say, "Oh, obvious." Now that I understand, it's obvious. So, but sometimes you need to model, and then you run many simulations until you understand the system, and then it becomes obvious to you after days and days exploring the system. So give me a thumbs up when you are done with this. Niraj's thumbs up. Fien's okay. Okay, let's keep going. So, boundary species. What is a boundary species? A boundary species is something that you want in your system as a constant, as a reference value. In biochemical networks, that would be, for example, ATP. You are modeling translation of proteins, which is very ATP intensive, but you don't care about your ATP loss because you don't want to model a huge metabolic network to regenerate ATP. So in this case, you say, okay, ATP has a fixed concentration, or maybe you can increase the complexity a little bit by saying, I do lose ATP, but I have a constant regeneration of ATP. So these are your boundary species. And setting a boundary species in the first approximation, so let's say I'm fixing my species at a constant value. In Tellurium, you do that by using the $sign. $S means Tellurium will not update the value of S. You have to set that to all reactions you are using S. Otherwise, it doesn't work. And let me reset this. So what do you guys think will happen when I run this? By fixing S at a certain value. This certain value is 1. So now I'm fixing S at 1. What will happen? Will something weird happen?
E and ES exponentially converge, and you'll get that to a quasi steady state, and
Yeah.
well, actually to the steady state.
Yes.
And P will just increase quasi linearly. We'll say quasi.
Forever?
Yeah.
Forever. Or not?
Since S is fixed, yeah, forever.
Forever. Okay, so Nick is saying that P will grow forever. So P goes to infinity. Your hypothesis. So what do you guys think? Do you agree with that, Niraj? P goes to infinity? P is unbounded?
I guess since you are adding substrate continuously to make it a constant value, so P would constantly increase. So yes, I guess.
Do you agree, Shen? Shen? Chen?
I have no idea.
You have no idea. All right. So in the previous cases, we were losing all our S and gaining all P. All right? So a full conversion of S to P. Why do you stop producing P eventually? Because you lose all your S. Again, this is a sophisticated case of S going to P. S goes to P. So when you lose S, you stop gaining P. But now you say, no, I always have S by using the dollar sign. I always have S, and S is going to P, but I always have S. So basically you have a constant generation of P forever. I hope this simplified model helps. So when I do this, that's what I have. So the hypothesis is correct. You get a steady state for E and ES, as you mentioned. And then you have a linear growth of P forever. So B is unbounded. This can be very bad in the model when you have an unbounded species. You can start having numerical problems. And your simulation breaks. So we have to be careful about this. So what is a way to fix this? Let's say I want to make P a bounded species. What is the first simplest possible solution for that? Okay, what do you think, Niraj? Or Chen, what is your idea to fix this problem?
Maybe like the second reaction where the complex of E and S converts to E plus P would be reversible. That way we could attain a equilibrium value of P.
I will do that and let's see if that works. What do you think? Will that work? He said that we should make a reversible reaction from E plus P to ES. Does that solve the problem? No, I don't believe this. Wait a sec. Wait a sec. Oh, but I think okay. So let's try R for R reverse and say this is zero. If that works, like you're saying, I've never seen this. Wow. So the reason for that is that now S is working both as a source and a sink. When you convert P, when P is very high, you start converting P back to S because it goes all the way back. And then because you're fixing S at 1, S becomes not a source anymore, but actually a sink.
Yeah.
Wow. That's very interesting. I thought it was going to fail. Hmm? Yeah. Well, I guess that's yes. But and I guess that's the reason why we do modeling, right? because sometimes we expect differently. Okay, so good. Good fixes. However, Niraj's gas is not adequate because we are changing the model structure. We are changing the way that ES interacts with E and P. This is a problem because let's think, for example, about the kinase case. So the kinase goes to a protein and phosphorylates it. And then when you do it reverse, you're basically saying that the kinase also has the capacity to remove that phosphate group from the protein. But if the phosphorylated protein is much more stable than the non-phosphorylated protein or the enzyme protein complex, then you don't have this reverse reaction, right? So basically, a kinase cannot do the reverse. It has to be done by another molecule. And then, so here you're changing the model structure. If you don't want to change the model structure as much, you do what Nick suggested, which is basically, oh, we are not considering that the protein P is degrading. So then you do this, D times P. Now you have a degradation for your protein. So if you set your degradation any value, you will have a limit for your protein P. So that's the simplest fix because you are not changing the assumptions of your model structure. You are just considering that P is also a protein, is inside a cell, it has to be degraded. Unless it's an amyloid fibril. In that case, it's more complicated.
Well, it just be things cell or whatever your sample is.
You can also use the decay as to model the secretion. So I guess, yeah. So you model it the same way, but you can interpret as degradation, secretion. What else could it be? I don't know. Or formation of other complexes in other processes. Okay, let's keep going. I hope this is the result. And then I was not expecting this slide. Oh, okay. Now I got it. Okay, so if you see this diagram is not explicitly telling you anything about S and I. So here, in this reaction we are seeing, you are explicitly telling something about S. But in this case, you're not saying what is happening with S. So in this non-complete notation, you will assume that S is a boundary species and I is a boundary species in such a way that you have the back and forth binding and unbinding of S with E and I with E. So why would you write it this way? Well, because sometimes you have those huge networks, those very complex networks for enzymatic reactions, for example. And you don't want to write oh, I have this boundary species, it binds to this and that. So these simpler notations make it easier to read a diagram. But of course, as someone looking at this in a paper, for example, and you think to yourself, what is S? What is I? well, they are boundary species. That's at least generally how we like that's our convention. So in the lack of information about S and E, you are basically saying that S is constant and I is constant. In this case, you have a constant production of ES that depends on E and depends on the amount of S you have, and also a constant production of ES that depends on, not constant, but you always have a regeneration of EI, let's say, which depends on how much E you have and how much I you have. And I is constant. So here the total equation, the complete equation for E, you'll have to account so this, I guess, this is let me see these again. Here I'm not quite sure why it's not E times I and E times S. So I guess we'll have to come back to this later with Dr. James. All right, now we want to simulate this system. So let me go back to this guy. We simulate this, or we already have simulated it. And now, to plot our species, we use, so for example, our result is this. And then we can select what we want to plot. So for example, time, and then your selected species versus time. So for example, ES and P. And then to plot this, to plot only the columns you are selecting, you do plot array. Result. It's TE plot array. And this way you just plot the species you're interested. All right, so the content of the class is over. So I have two more things left that I want to talk. So first is Niraj and Shen. Did you read the paper? I know you weren't here last class, so you didn't even know that was happening. Did you read the paper?
No, I forgot the paper, right?
Okay, no problem. Shen, did you read the paper?
Yeah, the paper about like using three substances and they
Yeah.
inhibit each other.
Yes.
That's the paper.
The
Yeah, I
recursive.
read about that. Yeah.
Nice. Okay, so This is one of the homeworks I will just show to Nick. Where is this one? So last class, we talked about gene regulatory networks. And this is the first ever paper about the repressilator. The repressilator is this thing over here, where you have a sequence of inhibitions. Last class, we talked about inhibition. Are you familiar with that? So are you familiar with the Hill function? Hill function? No? So very simple to you. If you want to say A inhibits B, you are basically saying something is producing B and A is inhibiting production of B. How is the ODE for that? So you have B prime is, then you have a constant production of B, but then A acts as an inhibitor, and the inhibitor is the more you have of that, the lower is the rate of reaction that you are inhibiting. So the Hill function is basically a function on this form. You have a half max, and then you have your Hill exponent, and then you have your half max to the Hill exponent plus A to the Hill exponent.
That's add term.
No, it's multiplying. Yeah. If it's added,
Oh, okay.
so to make your physicist mind work better, this function is like this. It's a sigmoidal function. It starts as 1. It starts at 1. When A is 0, it's 1. At half max, you have half. When A is equal to half max, you have 1/2. That's why it's called half max.
Yeah.
And when A goes to infinity, 0. And the exponent n regulates the steepness of this shape. So it's like a hyperbolic tangent or other sigmoidal functions you saw out there. So this paper talks about the system of cascade repression, inhibition, I should say, not repression, this cascade inhibition, which produces oscillations for certain parameter sets. And then he built a bacteria. So he made an experiment where he built a bacteria that contains genes that repress each other in this fashion. And then he put in the plasmid of the bacteria a GFP protein. The GFP is basically a protein that glows green in the microscope. And he made one of these genes inhibit the production of that protein. So basically what you have is this cascade inhibition produces oscillations. And when you have a high TET-R, you have a low GFP. So basically you have a oscillation of brightness of your bacteria in the experiment. Good. And then he used the model to understand what is going on with the system. And basically the model, because using the model, you can see what sets of parameters lead to oscillations. You can tell the experimentalist to say, change this variable, so change the concentration of this thing to produce stronger oscillations or inhibit the oscillations. So they made a very cool study about that. This is the experimental result. This is the oscillations produced by the network. And they also they make considerations. For example, what if the system is stochastic? Because it is stochastic in nature. So what happens if we make the system stochastic? Does it behave more similar to what I'm observing in the experiment? All those kinds of considerations. This is their network. So very simple network. This is your inhibition term. Do you recognize it? You have P is your protein. This is a Hill function. Your half max is 1, and you have an activation rate, alpha. So here you have your P to the N. And then, well, we talked in the gene regulatory network that your gene can be leaky. And it talks about leakiness of genes. Here he gets to the stability condition for the system. Discussion about what are the effects of making the system stochastic. And that's it. It's a 4 pages paper with 6,000 citations. So this is the paper I chose for you guys to read. And these are the questions, which I have my answers here. By the way, the questions, I didn't read the paper before making the questions, so I made the questions, and then I read the paper to answer the questions. So the first question is, what is the objective? What is the problem and question they want to solve? Describe the structure of the model. What are the nodes? How do they interact? Does it conserve mass? Is it gene regulatory, protein signaling, metabolic, or another type, which we already know now? What are the simplified assumptions they make? What are the measurable quantities they use to validate the model? And what are the conclusions? And after you read the paper, so hopefully next class, you guys will have read the paper, we'll have these follow-up questions because the paper is interesting. And when you want to understand the paper, these are the questions that will probably pop up in your minds. What is low leakiness? What is the parameter in the network that corresponds to this leakiness of the gene? The author mentions an obstacle to the design of biochemical networks. What is the obstacle and what strategy the author used to circumvent it? So these are considerations. This question is important because if you in this class will work with or build a network to work with, you will face this. You'll face this question. Like that obstacle that the author is talking about, you'll face it. And he is presenting a strategy to dealing with it. What is the importance of IPTG to the experimental setup? What is IPTG doing? What is the effect of introducing stochasticity on the behavior of the network? These are the follow-up questions. This will answer together next class. Okay, so you don't. This is not a homework. This were here. It's not a homework. So don't worry about answering this. I'm just outlining what I will do in the beginning of next class. Okay, just for you guys to not have any surprise about this. And what did the model provide to the authors of understanding of the system that they will not be able to know only from the experiments? That's the final and perhaps most important question of all. All right. Other than that, I have 2 suggestions by James to Shen, and then I will finish the class. So the suggestions go like this. So let me open my whiteboard again. So optimization of networks. This is something that Shen is interested in.
Yes.
So optimization of networks, there are 2 concerns currently on optimization of networks. that James is particularly interested in. I am also particularly interested in. And if you are interested in one of them, then you can make a project in this class about it. And you can also include your other teammates. Like you can, by the way, you guys can all do the same project. That's not a problem. So the first idea for optimization of networks, this one is simple to explain. So in networks, you have one optimization problem, which is you have your nodes, blah, 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 like this. And then all of these arrows have parameters, sometimes 2, 3, 4 parameters. So let's say this network has like parameter K1, K2, blah, 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 Kn. All right?
Mhm.
And then you have your data. So you have, for example, let's say this is node A, this is node B. You have your data. This is A, this is time. You have this, and then you have another data for B, so on and so forth. So given the data, what are the parameters that better fit the data? That's the most basic optimization of networks problem out there. So you have your network, you know about your network, you know the parameters, you have the data, find the best parameters that fit the data, right? We are probably all like aware of this problem, like linear regression, whatever.
Yeah.
But then there is a problem. And you saw this problem in the paper, which is the guy that built that repressilator model for his bacteria model, he is not sure whether that model he created is accurate or not. Those inhibition functions he was using, or whether it is an inhibition at all. Isn't it the case that actually one of the protein was degrading the other protein or binding to the other protein instead of acting over the RNA, is acting over the gene regulatory part? So what are all the simplest types of networks that fit the data? So you can have, and I'll give this an example. He has this, but could it be just this? Could it be something like this? You know? So can you achieve the same network behavior and optimize your model to the data with all those various structures?
Okay, I understand. It's like a reasoning problem. Like you have the data and you want to find the minimum or the smallest size of model that you
Yeah,
can.
that's one of the questions you can ask. But you can also ask, for example, how many of those combinations are successful in achieving the intended result? Or for example, I have a huge network. What is the smallest? That's the one you're asking about. But this is a different kind of optimization problem, because first, you cannot use that regular gradient descent optimization, because what is the gradient between this and this, right? There's no gradient here. I mean, probably if you are a good mathematician, you can come up with some generalization for gradient, but uh, I am, go for it. But that's I will not be able to help you on that. So this is a discrete optimization, which involves changing the model structure. And inside each model structure, you have to optimize the parameters too. Because how do you know, let's say, I fixed the parameters and I changed the structure and it didn't work. But okay, you changed the structure. Now you need new parameters. So by using the structure, can you find parameters that achieve the same result or not? So for all of these combinations, you have to optimize the parameters to see if that works or not. There is a second concern in terms of optimization of networks, and this is very cool. And the second one is something that real models face. Like you build a model, let's say you build a model for pandemics. So you have your SIR model, and then the pandemics is going on. And then you have that curve, like how many people are infected. And you want to use that data to update the parameters of your model to make predictions, to see how it's behaving. But you have limited data. And you have like one set of data, noisy data, to fit 10 parameters. So it's impossible to do a full fit. In this case, let's say you have a simple model. Let's say you have 3 parameters. You have K1, K2, and K3. You have 3 parameters, but you just have one curve. You just have one shitty curve like this. Then in your parameter space, you cannot do a proper fit. What is a proper fit? A proper fit is a region in the curve in your parameter space. So you have your solution space inside you have your solution, and there is an error to the solution. So you have an error. So it in all your 3 parameters. So it's a volumetric uncertainty region in your parameter space. All right. And this uncertainty region decreases as you get more and more data, right? So the problem here is, I give you a curve. And this curve can only give you one constraint. Let's say this curve tells me that K1 is 2K2. If you have this, you are limiting your solution space to a plane in your solution space. So all these infinite possible combinations of parameters in this subspace solves the problem, fit to the data, right?
Mhm.
When you have these limited knowledge and this huge set, this infinite set of solutions of possible parameters for a system, but you still need to make a decision. You still need, for example, you have a patient, the tumor is growing, etc. You need to make a solution using your model. But you have infinite possible combinations of parameters. Then what they do is pick random combinations of parameters in this set in this solution set, in this solution space. So let's say I picked by chance, I picked this one, this one, this one, this one, this one, this one, this one. And so I picked all these parameter sets. And then for each of these parameter sets, I make the question, what will happen when I apply a certain drug? What will happen if I start the lockdown now? What will happen if people start using masks now? What will happen if the virus has a mutation now? But because you have all those models, now you're asking yourself, in how many of these models does my solution work? So instead of saying this solution, this perturbation of the system will lead to that result, you'll say it will lead to that result in 60% of my parameter sets. And in the other 40% of my parameter sets, it leads to catastrophic results. So in this case, the risk is too high, I will not use the drug. Or in 50% of the cases, it didn't do anything. And in 50% of the cases of my simulations, it was a little bit better. Well, in this case, I might consider using the drug. So even with limited data, you cannot specify the parameters of your model. You have an infinite space of solutions. You can still use this method of picking random parameter sets in this space, in this parameter space, and give me a probability of success of a certain intervention. Is that clear?
Yeah, it's a random sampling. Yeah.
Exactly. So you are sampling models. And
so what to optimize here?
So what you optimize here first is with limited data, optimizing your parameters, which is basically this, right?
But if you are doing random sampling, the quality, it doesn't matter what the values are. What's the criteria of your optimization?
That's a good question. That involved, like it's contained in the project. So that's part of the problem. I don't know the answer. Like I
Okay.
don't know how.
Yeah. So the first option is quite interesting and clear, but the second one it seems to be just a random sampling of models. So
Okay.
I don't get it. I don't know what to optimize here.
Yeah, but yeah, maybe it's not not a huge optimization problem. Yeah. Yeah. Well, the first one, it's a
Yeah, the first one is indeed a huge optimization task. Yes.
yeah. Well, I'll leave that for you guys to think. Do you guys have any questions? Anything else to say? If not, you guys are free to go, and I see you next week.
Oh, okay. See you.
All right, goodbye.
Bye.
Hayden, you want to talk about it, or
Only if you do. Did you get is the other student there in the class still?
No, he's out.
Did you get his name?
Nick.
Nick. Last name or just first name?
I just know the first name. It's
Okay.
Nick.
Oh, that's promising. I guess we have 3 people that are taking now.
Yeah. Yeah.
Yeah, I mean, I don't have too much feedback. I think that it went well today. People were engaged. I like your
Yeah.
more one-on-one teaching style, and since we only have a few people, we can actually afford to do that this semester. So
Yeah. I didn't have time to change the slides. So James sent me an email Sunday say, oh, you need to see the slides.
Mhm.
And then I said, ah, okay, I'm really teaching next class. So then I got his slides, I just like shuffled it and
Yeah, I got your email yesterday about it. Didn't have time to take a look before today. Things have been pretty busy. Um, yeah, I think it's going well. I had to step away for a few minutes at the end. I came back halfway through that optimization question. What was the student asking? I'm trying to remember his name.
who? Shen, the Chinese?
Yeah, let me find. Wrote that his name earlier.
It's Shin Xiao. It's like
Shin Chao, yeah.
Shin Xiao. And then I think it's Fa Fu, whatever. Fa Fu
Ooh.
Fame.
So what was he asking about optimization at the end there?
No, so those are he